Hey, thank you all for being here. Also, hello to the people online. It's a pleasure for me to present you to Thomas Zajkowski. He was born in Warsaw, where he studied genetics, but he's spent the last 7 years in US, mainly in Stanford University and in NASA Ames Research Center, working in many different subjects, but mainly focusing on prions and their relationship with the tree of life. And actually, this is the talk that the subject he's going to talk to us today. He even created a startup at Silicon Valley last year. So I guess he should be multimillionaire by now, or almost. Not yet, but soon. And he loves astrobiology, actually. And he founded a few years ago the Polish Astrobiological Society. So even he's quite young, he has done lots of things everywhere. He has decided to leave US and come back to Europe. And so he's in a critical transition of his life now. He doesn't know whether to go back to Poland or perhaps to another country of Europe, but his idea is to create here a new group on astrobiology. And so all connections will be great for him. So if any of you want to talk to him afterwards or have lunch with us, that will be great. Otherwise, you can contact him whenever you want. And well, I think that's all. Please, Thomas, go on.
Thank you so much. Hello, everyone. I'm super excited to be here. I always wanted to visit this institute. If you don't mind, I'm going to sit so I can see my notes better. Okay. Thank you, Jacobo, for your kind introduction. And as I mentioned, I'm really thrilled to be here to meet all of you. I love Spain. It's my first time in Madrid. I'm already loving the city. I found this comparison. I think it's like a fuse of Warsaw and New York. If you just take it and make an average, you get something beautiful. So today I will be talking about the relatively little known mechanism of cellular regulation that is recently gaining a lot of attention in the scientific community, and it is the prion-based mechanism of regulation and adaptation. I will also talk about other self-templating protein aggregates called amyloids. And many of you might actually recognize the name amyloids in association with Alzheimer's disease, at least. But I will talk not about the Alzheimer's disease, but the role of amyloids in microorganisms, as well as their hypothetical role in the origin of life. I'd like to give you a little bit more background about myself to better understand where do I come from. So as Jacobo said, I am a scientist. I'm mostly interested in evolution and of the universal processes such as genetics. I'm also deeply motivated by the potential of synthetic biology. How can we use synthetic biology as a tool in space exploration? I did my master's in genetics at the University of Warsaw, followed by PhD in neurobiology in the Polish Academy of Sciences, and where I studied molecular mechanism of the prion disease. So I got a training in neurobiology pretty much. Toward the end of my PhD, I discovered the incredible research area of astrobiology, and with that, I found my passion. So pretty late in my life, I found the passion of astrobiology, and I bet you'll share the same. As many of you, of us here, I want to answer the most significant, as I think, questions about our place in the universe. Where do we come from? How did we evolve? And how can we enable our sustainable future as a spacefaring civilization? I am currently affiliated with the University Space Research Association, working at NASA Ames Research Center. I spent 6 years there working in person with Lynn Rothschild. Right now, I transitioned to a remote work supervising a project. My research focuses mainly on my individual project that is concerning the evolution of prions. But I also helped with several other projects happening in the lab. That includes myco-architecture, which is trying to use mycelium as a functional construction material of world, so on the moon or on the Mars, to like bring together regolith and make structures of it and give it a functionality of biology, because it's a living system. So we can try to do that. Another big project that got several rounds of funding of a NIAC, so that's a NASA Advanced Concept, and Lynn is really good in getting those grants. It's astropharmacy, where we use biology to synthesize drugs for astronauts. And currently, I'm working remotely, as I mentioned. I'm supervising a project with Mark Ditzler and my student, Max Packebusch. And the project is titled The Role of Amyloids in the Earliest Forms of Life. So it connects to the origin of life. Mark Ditzler is an RNA biologist. I also had a pleasure to work with Ivan Paulo Lima. Maybe some of you know him. He developed a successful space mission, PowerCell, that's the name of a mission. It went on a satellite called Eucropis. It was a German satellite, German mission primarily. And he showed for the first time that you can do bacterial transformations on a satellite. So synthetic biology in space. I'm also a member of Blue Marble Space Institute of Science. I hope some of you are members or are at least aware of that. And SaganNet, that's a nonprofit organizations that gather astrobiologists and educators. Very vibrant. They have very active Slack, and they're also one of the contractors for NASA. So if you get affiliated with them, you can get hired by NASA then. I also have experience in an industry, as Jacobo mentioned. So 1 year I helped funding, creating a Silicon Valley. I'm not a CEO, I was a funding scientist. It's called Switch Bioworks, and the goal of the startup is to use synthetic biology to create tools such as biofertilizers to fight the climate change. And I think personally, in an astrobiology context, I think that biological fertilizers will be necessary in the future of space exploration, because no one is going to bring heavy chemical fertilizer with them from Earth to space, or produce fertilizers in space using a Haber-Bosch process that requires like 400 atmospheres and incredible pressure, 400 atmosphere pressure, and like incredible temperatures. Establishing such a facility in space would be extremely challenging, while instead we can use bacteria to do exactly the same, produce ammonia in normal room temperature and pressure. So if we ever imagine eating vegetables on Moon or Venus, I think, in my opinion, the biological fertilizer is necessary. And of course, the biological fertilizer have obvious benefits on Earth, specifically in light of climate change, like greenhouse gases emission that it produces, ozone layer depletion, or water pollution. And last but not least, I'm the founder of the Polish Astrobiology Society. We started as a group of just science fiction lovers and discussing them, and we quickly realized that a lot of us do fascinating research in astrobiology and started giving talks, bringing people together. Right now, it's about 100 scientists on board. I'm still running it like it was 10 of us, so I need some help what we can do with it, and collaboration is always useful. One of our main goals is making astrobiology accessible and inclusive. And that's why we organize every year. This year is going to be our third annual International Astrobiology Conference. To keep it cheap, we make it online, and it will take place in December. And some of you already took part in it. Thank you very much for your contribution for the previous iterations of it. And I invite you to invite your students, and students, please come and send abstracts. We just extended the deadline for another week. So you have 1 week to apply. I will show another slide about it at the end, and you can get a QR code from there. Yeah, so thanks so much. And as Jacobo mentioned, I'm moving back to Europe and I'm trying to establish my own research group. And with this, I will be starting the main part of the presentation. And my main presentation focuses on prions. Prions are fascinating. And they're fascinating because of many reasons. And one of them is that they're super controversial. In the course of the talk, I will give you several examples why they are controversial. But let's start with explaining the name itself. The term prion comes from proteinaceous infectious particles. So in other words, it is infectious particle built from just a protein. So prions were first discovered as a potential as a protein aggregates in patients suffering from infectious brain disorders. At first, the infectious agents were thought to be something more classical, like a virus, but they were called slow viruses because it could take decades to develop the symptoms. And it also took decades for the scientific community to accept the controversial idea that the protein itself, without any nucleic acid component, can carry biological information, and a consequence, developing a phenotype that in this case is a disease phenotype. So another controversy about prions, this controversial subject is associated with cannibalism. So in 1952, a Nobel laureate, Daniel Gajdusek, traveled to Papua New Guinea, where he started his research on something that seemed to be a first infectious neurological disorder. The disease was called kuru. In the native language, the kuru means to shake. So infected individuals were suffering from ataxia. They had problems with holding a straight up posture, and they had tremors, and that's why they used the sticks to support their straight position. Interestingly, Daniel Gajdusek, he noticed that the disease affects mostly women and children, and later he found out that those tribes performed cannibalistic rituals, and to honor their dead, they would eat the parts of the bodies of their relatives, deceased relatives, and while male warriors would eat mostly less infectious muscles, women and children, they were eating brains, and that's where the infectious material is mostly located. That's why they got more affected by the kuru disease. And then later in the 90s, prions became way more recognizable to the general public because of the epidemic of mad cow disease. And probably everyone here heard about mad cow disease. And it broke out in Great Britain. They had to destroy millions of cattle to prevent the further spread. And it severely harmed the economy of Great Britain at the time. Another controversy about the prions was that it seemed that it jumped from species to species. It could cross a species barrier. There's not many diseases that can do that. And precisely it was spreading from cows to human. And now we know that a similar diseases exist in very different animals. And one of them it happens in wild deer in the US. It's called chronic wasting disease. And it's spreading right now in North America in 2023. So it's a common issue even right now. So prions are not gone in the context of pathology. But the molecular mechanism of the infectivity was finally explained by Stanley Prusiner, who was awarded a Nobel Prize. He discovered that infectious agent is actually smaller than a virus. This infectious agent could survive treatment that normally destroys DNA and RNA. It turned out that the infectious agent is composed solely out of a protein. So Stanley Prusiner named this protein, he called it a prion protein. And both Daniel Gajdusek and Stanley Prusiner were awarded Nobel Prizes for studying prions. And another maybe last controversy here that is not directly connected, it's not connected at all to scientific merit, is that a Nobel laureate committee, like a Nobel Prize committee, decided to take the prize from Daniel Gajdusek because he was convicted for child abuse. And as you can see, the press around the prion is very bad. Even though scientifically they're fascinating, there is a lot of bad press around it for some reason. So prions are still the only one infectious agent that has no nucleic acid component discovered. This difference makes the prion replication itself fundamentally different from all biological processes that we know, and that's what I find the most fascinating about prions. the fact that the prion can store biological information, it seems to contradict our understanding of the information transfer in biological systems, something that we call the central dogma of molecular biology. But on the other hand, this unique behavior promises some amazing discoveries in the fundamentals of how the biology actually works. But only now we start to begin and to understand how important the biology of prions is in a physiological general phenomenon of life. And that's what this talk is going to be about. So for the last 20 to 30 years, prions were widely recognized as infectious proteins. But recently, we have started experiencing a dramatic shift in the perspective on the prions. They come from very rare disease-causing agents to common biological phenomena. So recently, prions were found not only in mammals, but in many other organisms, including plants, but also in microorganisms like bacteria, archaea, and even in viruses. There are prion-like proteins discovered in viruses. What could they do there? Even more interestingly, prions were found to have normal physiological functions. So they're not necessarily pathological, especially in microorganisms, they play normal physiological functions, but even in animals. I'll give you a few examples soon. So to better understand this whole phenomenon, let's first take a look at some molecular biology, molecular mechanism of the prion replication. And I will start this animation and works. So this animation shows you the formation of the original prion that causes the mad cow disease. So we're looking at prion protein, the first prion protein discovered. There's an overlap in naming that is sometimes confusing. This is a prion protein, that is a prion protein. At the beginning, the prion protein can change its secondary structure, increasing its beta sheet content. That's what you saw. And then it misfolds into a protein that is interacting with other copies of the same protein, changing it into the same conformation that is reaching beta sheets. And it starts building on top of each other in the form of a fiber. The process repeats until all available subunits are depleted, and it's exponentially fast. So the aggregation is a building of a higher order structure that grows in one dimension, forming a fiber. And such fibers are shown on the right side of the slide, and they're called amyloids. So this is the first time in this talk when I start talking about amyloids and the overlap of amyloids with prions. So the typical shape of a prion aggregate is called amyloid, but not all amyloids will be prions. So most known prions acquire the form of amyloids. Thus, the prion may also be seen as a subtype of an amyloid, as I just said. And importantly, amyloids that are built out of the same protein can take slightly different shapes. So you can start with the same protein that aggregates to combines together, building higher order structures, but at each time it's going to build the structure a little bit differently. So the shape depends on the final conformation of the protein because it can misfold in different ways. The fiber is a little bit different. And interestingly, the different shape of a fiber have a different impact, for example, on infectivity or the ability to cross species barriers. So the same protein is going to have different capability of crossing species barrier as an infectious agent, depending on what conformation it acquired. The conclusion is that the biological information is stored in the structure of the amyloid itself, of this fiber. Oops. So this graph shows you different stages of prion formation. First, reversible oligomers are formed, leading to a formation of a protofiber. And the protofiber starts elongating exponentially quick. And then eventually, available components are depleted and the aggregation reaches a plateau. This is important to remember that aggregation is quicker than the synthesis of the protein, so it can lead to the depletion of a functional protein within the cell. From the protein that is the subunit of the fiber can get depleted. So similarly to prions, amyloids are, the term amyloid itself is generally recognized in association with disease. And these are some examples of the most recognizable amyloid-related diseases. So we can find those fibers in those diseases, but it doesn't mean they're infectious. Only the prions would be a subtype of infectious amyloids. But also amyloids, we look at this list and we think, okay, there's all disease, but there are not necessarily bad in other examples. And it comes from its ability, its very nice properties. The mechanical properties of amyloid itself can be very useful in nature. So they're very stable in the wide range of temperatures, pHs. They have a very high tensile strength. So it shouldn't be surprising that the nature is using it. Those properties are utilized in many processes, such as storage, scaffolding, building incredibly strong fibers, such as silk or spider web. Amyloids are a component of those, and even in biofilms. But amyloids are utilized not only because of their mechanical properties, so the tensile strength, for example, but also because of their ability to quickly aggregate, and in consequence, causing massive changes in the protein availability in a relatively short time. So this behavior it was discovered to be essential, for example, in an immune response. Ability of a protein that takes part in immune response to aggregate is necessary to create the immune response. Or long-term memory formation, the protein ORB2, and that is conserved protein in mammals, but also in Drosophila. If you remove the part that enables aggregation, it will influence the ability of those organs to create long-term memory formation. So typically physiological process, not pathology at all. From the space biology perspective, the research on amyloids might bring solutions, for example, to the serious problem that is called by biofouling. So biofilms formed by many species of bacteria cause problems not only in medicine, food production, but also in spaceships. There are several failures of a life support system, components of the life support system documented on different spacecrafts, including Soyuz or ISS. So a serious threat. The more people spend time in space, the more we have to deal with biofilms. And amyloids are an important component. It seems like they create a scaffold for the rest of extracellular matrix. And one of my grants that I recently wrote, trying to establish a new lab, is focusing on the role of amyloid proteins in biofilm formation. Okay, I have talked a lot about the prions and amyloids, and both terms seem to strongly overlap. So I want to emphasize again a little difference that is between those two terms. I'll try to explain it with this graph. So the formation of a functional amyloid, what you can see on the left side, is usually associated with gaining a function. So the protein is expressed in the cell, then exported and forms a fiber, and for example, helps with biofilm formation. While in contrast, the prion formation is often associated with the loss of function. So prions can also spread between different cells. That's important. And they're synthesized in a cell. And green color indicates like the functional form of a protein. But then it starts to form this oligomers, a fiber, and in the aggregated form, this protein loses its function. And the prion can be then released. It can be inherited. It can spread between cells. It can be shared from a mother cell to daughter cell, changing the whole population of cells in a consequence. Let's take a look at some common characteristic of prions or the prion proteins and see how can we try to discover new prions. So thanks to the development of the machine learning tools, it became possible to predict prion-like behavior in large data sets of biological information, such as protein data bank or UniProt. So the algorithms are trained on known prions, and they're used to create a list of prion candidates. The prediction is based on some unique characteristics of prion domains, so a part of a protein that is responsible for aggregation. And some of those characteristics are that the algorithm itself takes multiple different characteristics under account. So it looks at the complexity of the region, and it has to be a low complexity region in the protein. It has to be abundant in glutamine and asparagine. It has to be depleted in hydrophobic and charged amino acids residues, and there has to be a long disordered region present within the sequence that is being analyzed. And as you can see on this image, those little tails are the aggregation domains. So they're called prion domains, and the prion codomain is usually separated from the globular part of the protein that performs a function of the protein. So very interestingly, when you artificially attach such a tail to a different protein, it can convey the aggregation capability to another protein. So this modularity makes it relatively easy to create a new artificial prion. So it can be used in synthetic biology, for example. So another characteristic of prions is that their aggregation can be reversed. But how do you, so the prion can show up and you can then turn it off. How do you turn off the prion? To understand that, let's look here and start with the fact that prion can show up spontaneously. It is just a matter of statistics. When a protein with a certain amino acid sequence and a composition that permits the prion-like aggregation, it will eventually acquire the amyloid form in which it can spread. This behavior is usually balanced by proteolysis, so other proteins that destroy accidental aggregates, preventing toxicity. Protein aggregation is a general problem in a cell, and there is strong anti-aggregation systems. So prions are handled by those two. Prions can be also inherited in a cytoplasm or picked up from the environment. So when a prion-like aggregate is formed, it can be either disassembled to get rid of the prion. You can either disassemble it by chaperones, or it can become so massive and so stable that it's locked in the mother cell during division, and the daughter cell is free of prion, and the whole offspring of this free of prion cell for some time is going to be free of it until statistically one of the proteins is going to acquire this fold of promoting aggregation. So after spending 6 years in California, I could not stop myself from making a reference to the former governor of California, Arnold Schwarzenegger. And on this slide, I'd like to explain why prions are like Terminator, played by Arnold Schwarzenegger in one of his movies. So prions, even when destroyed, they will come back. And that is because of what I just explained on the previous slide. It's a matter of statistics when a protein like prion-like protein is going to acquire aggregation-prone conformation. And this is very nicely illustrated in the simple experiment done by Reid Wigner, which showed that if you look here on the top side, you see my cursor, great. So this shows that 1 in a million divisions, there will be a prion form acquired, and it takes only one generation to cure it. And then it takes another about 1 million divisions until you get another spontaneous formation of a prion. But if you overexpress the amount of the protein in the cell, you get more likelihood of creating a prion, and now it takes less generations, 2 orders of magnitude less generations, to get a prion phenotype out of it. Even when cured, the prion phenotype will eventually come back as like a terminator. And this observation brings us to the very important concept that phenotypically, prion can look like a reversible genetic mutation. But before I start introducing this concept of prion-based genetics, let's make a quick summary of what we learned so far. So far we learned that some prions can quickly form amyloid aggregates, that phenotypically such an aggregate can mimic deletion of the gene that codes for the protein, because now the functional protein is engaged in forming an aggregate and no longer plays its role. In case of prions, those phenotypes can be reversible. And how often a prion phenotype occurs and how long it lasts depends on the rate of expansion of the gene, the rate of expression of the gene, so producing of the protein, and the rate of degradation of the protein or the protofiber before it creates the stable heritable aggregate. And that brings us to the conclusion that prions behave like heritable molecular switches, and the rate of on and off state is evolutionary balanced or tuned, if you like. So this concept here, I'm just showing 2 striking phenotypes when the prion is off and a prion is on. And in a cell level, you can see that prion forms those foci, little aggregates inside the yeast cells. And prions are best studied in yeast so far. So the concept enables explanation of the concept of prions after Stanley Prusiner allowed scientists to explain very weird phenotypes in yeast that were observed since 60s and 70s. So this protein inheritance helped explaining why these phenotypes show up and disappear so randomly. And here you can see an example of those phenotypes, red and white. It's very easy to track. And these are the models that are used for tracking the influence of prion, for example, on adaptation. And this slide explains how this adaptation could work on a population level in regard to prions. So from the population perspective, a prion-like switch can lead to adaptation to functional, to like fluctuating environments. And those mechanisms I described as something that is called biological bet hedging. And the term bet hedging itself comes from gambling. It's when a bet hedging is when a player puts a very small bet on a very unlikely outcome, but if it succeeds, it gives you can potentially generate a big reward. And in case of microorganism, the reward is a survival of a whole population and a genotype. So on this graph, on the top of it, you have environment number 1, and on the bottom, the blue one is environment number 2. And we start with a population without a prion formed in any of the cells yet. It divides normally. By the way, this is how geneticists write prions in small letters and brackets and minus. That means it is a prion non-active state. And then one of them becomes red phenotype, as you saw in the previous slide. And this population now is diverse on a phenotypic level, but it has exactly the same genotype. Just one protein is aggregated. And when the environment changes, it might be that it's not permeable, it not supports the growth of all the cells except the one that has a different phenotype because of the prion present in it. And it is free to then rebuild the population. In the meantime, the classical genetic mutations can happen, and the cell eventually can lose the prion, but be able to sustain the environment number 2. So what happened now is an adaptation, and the prion is no longer needed. It's a burden for the cell to carry a big aggregate, so it disassembles it, and eventually the population is recreated with these new mutations that allow it to survive the environment number 2 if you encounter again. And to me, it looks a lot like Lamarck. It's an inheritance of experiences. So from the adaptation point of view, a perfect prion is a protein that, when turned into an inactive state, releases a cascade of changes that in consequence strongly alter the phenotype of cell. And if we look at the list of the best studied prions so far, we find that those are the typical functions of prion-like proteins. For example, translation terminators. The effect of aggregation of a translation terminator have effect on translation of every protein in the cell. Transcription repressors that regulate thousands of different genes. RNA binding proteins, another global function. And maybe the most striking is the chromatin remodeling factors, and that's one of the prions called SMAUG in yeast. If we look at the term itself, the epigenetics, in Greek, it means that it is outside epi of genetics. So something that is being on top of the classical genetics, like chromatin remodeling. So in this case, if we have a prion that is a chromatin remodeling factor, it's a epigenetic mechanism that regulates another epigenetic mechanism. So it's really on top of what is going on in the cell in this case. And here are some few quotes from the researchers on prions. So prions seems to integrate diverse inputs and actuate different outputs, provide immediate access to genetically complex traits, and because of their functions that the proteins have before they aggregate. And my mentor, Daniel Jarosz, he says that controlling the protein organization using a prion-like behavior is a new paradigm in biochemical regulation on a single cell level. So it's a great tool for synthetic biology. And in recent years, we witnessed this shift of perspective, as I just showed you, from the rare infectious agents to common proteins with important physiological functions. But I have one more question that is very astrobiology relevant. How old are the prions? And how old they are, I think, to help answer this question, we should ask ourselves, like, how old are the amyloids that are the functional shape of the prion aggregates usually? And because amyloids constitute this heritable form of prion. And here are some arguments that amyloids could be very old. So they're found in all domains of life. The beta sheet is the basis of it. And we know that beta sheet structure is one of the ancient ones. If we look at the structure of ribosomes, the oldest part of the ribosome are beta sheet made. And amyloids are also the lowest energy state that the protein can get on the folding landscape. And they can be formed out of very short prebiotically possible peptides like this one. So the amyloid fibers can be formed out of amino acids that are found on meteorites, and even from very short peptides like dipeptides. And so there is no limit on the length of the peptide that can form amyloid. So these are the kind of questions that we are trying to answer at NASA Ames. We think that amyloids could be the first enzymes. So the modern enzyme that comes from the dilemma and the problem in the origin of life, that the modern enzymes need hydrophobic cores to catalyze the hydrophobic pocket. And the protein to create a hydrophobic pocket has to be at least more or less 80 amino acid long. But to make 80 amino acid long polypeptide chain, you need a ribosome. But the ribosome is too big to self-assemble from prebiotic components. So what was the catalysis before the ribosome? Some researchers showed that mineral surfaces can be catalytical, but maybe we can have some biological catalyst too. And amyloids were recently found when they're formed out of short peptides, and we know that amyloid can be formed of something that is even shorter than 10 amino acid long. And so researchers find more and more catalytical functions of those amyloids made of short peptides, like ester hydrolysis, aldolases, ATPases, carbonic anhydrases, a copper-mediated oxygen activation. But most of them are lysis, like something that is not helping with building higher order structures, what we really want in the origin of life. So at NASA Ames, we're focusing on RNA ligations, and we have some concepts of experiments, and slowly moving forward to that. Right now, we are using, to answer those questions of amyloid's role in the origin of life, we're using cell-free expression system encapsulated in liposomes, and that allows us to enable selection and evolution of DNA sequences that code for short peptides, that can build amyloids, that can potentially be catalytical. And we have some ideas how to fish out those liposomes that have the catalytical activity. And by creating, for example, RNA ligation, you just do a reverse PCR and you find out that some of them have a longer product. Then we can come back to the sequence of it and learn about the DNA sequence that coded for amino acid sequence that eventually created the catalytic amyloid. I'm happy to chat more with interested people after the talk. So the example on the previous slide was how we can study the ancient amyloids, but what about the ancient prions? So in this case, in my study, I'm using a top-down approach. So at the beginning of my project, when I was looking through the literature, I found out that prions have so far been verified and discovered in eukaryotes, and only 2 of them in bacteria, and none in Archaea, and that was 3 years ago. So with this little number of examples, of prions, it is impossible to draw conclusions about the phylogenetic relationship of different prions. So my priority was to find more prions to be able to assess the evolutionary origin. So of course, the experimental verification is going to take more time. So we started with bioinformatics and used those prediction models that I described before. And we used this bioinformatics search performed on more than 1,000 archaeal proteomes, all available in UniProt, good quality proteomes. And we showed that this distribution of prion candidates on the phylogenetic tree of Archaea is more or less uniformal. Then we went to the lab and we concluded, we conducted a series of experiments to confirm the prion properties. And out of 16 selected candidates, 8 of them were able to form amyloids, and 6 of them were behaving like prions, or when we substituted a prion domain in a well-known prion phenotype in yeast, and they were changing those colors as expected. So in the first experiment, I used a method in which the candidate prion protein was expressed and exported by E. coli. E. coli grows on media. So these are just spots of bacteria on media that has Congo red. And if a amyloid forming protein is expressed and released, it changes the color of the colony. So it's very easy to track. And then we went into electron microscopy showing the fibers made by those proteins that we found in Archaea, used a bunch of biophysical methods like binding out to the amyloid-specific binding dyes, who show the kinetics that is consistent and typical for amyloid formation. And also we used infrared spectra and ATFR to show beta sheet content in the aggregate, eventually looking also on phenotypes of yeast when expressing those proteins. And in conclusion, our observation completed the picture of the prion distribution within the main branches of the evolutionary tree of life. So what I did in the last 3 years is I filled out this dot. And it looks like that our observation completed this picture, and the evolutionary tree of life is now full of prions, and it suggests that maybe there is a continuity between the first abiotic amyloids that could even take part in the origin of life and the modern prions that act as a regulators of numerous biological processes, and unfortunately sometimes contributing to pathological conditions such as prion disease, and that's how we discovered it. So we started from neurobiology, now we're going back to the origin of life. Well, they've probably been here since the origin of life. And the search for ancient prions continues. In our recent paper, we analyzed phylogenetic distribution of prion candidates. We now know that our prediction models have about 50% of success rate on predicting aggregation, so it's pretty high. So now we use the whole UniProt database. And as a reminder, prion domains are usually separated from the functional domain. So this time we analyzed what functional domain coexists with prion domain, what functions usually exist with the prion tail in the same polypeptide chain. And we specifically focus on prion candidates whose function can be seen across all three domains of life. So the function that could be conserved, and therefore we're looking for possibly the oldest prions on Earth. So we found two of the enriched functions coexisting with the prion domains that are conserved across all three domains of life. That's calcium ion binding and helicases. Both are central to signaling and regulation. And some of the candidates were already confirmed to form heritable aggregates when expressed in yeast, or are found in Drosophila and in fish as a potential candidates for prions. So that makes our predictions even more valuable. So our next step is to establish a high throughput method, a system that would allow us to verify experimentally thousands of possible proteins, prion-like behaviors in those candidates. And because we think that those candidates annotated with universally occurring functions are potentially representatives of the oldest prions on Earth. I'd like to finish with this quote because I personally agree with this point, and it's an opinion that the largest paradigm shift, the prions, as we discussed them, they represent the largest paradigm shift since identification of DNA as a molecule of heredity. It's pretty bold. And with that, I'd like to thank collaborators and thank you very much for listening. I'd like to also invite you for the Life in Space conference. We have some great guests there. Thank you so much for your patience, and I'm happy to take some questions.
Thank you so much for such an interesting talk. We have time for a few questions, so if anyone has one, just raise your hand.
Yes, thank you very much for your talk. Very interesting. I was particularly intrigued by one of the comments on your slides where you said that the prion or the amyloid was the lowest energy state. Okay, so in my understanding, if I can summarize a little bit what I understand from your talk, so the prion or the amyloid, okay, what it does is it steals proteins from folding correctly and giving a proper function, what you call an improper folding. But from the energetic point of view, it doesn't seem improper at all. It's the lowest energy state. So from a thermodynamic point of view, it's following the laws of physics and dynamical systems. So the question is, in normal organisms, because these prions induce and amyloids induce pathologies and diseases, which is something we don't want. So what is the mechanism? Is it known what the mechanism is in normal healthy individuals so that the folding avoids this lowest energy state? Which, by the way, is probably a non-equilibrium state, because as you know, the closer you get to equilibrium, the closer you get to death, right? We want living organisms have to be out of equilibrium. So has this study that you showed, which seems rather ambitious, that people have claimed that the amyloid is the lowest folding state, what is this based on? Some massive computational calculation of looking at folding versus energy? And then they concluded that these were the lowest energy states, or is it a hypothesis?
Honestly, I don't remember details of that paper, but well, let's take it out after the conference, after the talk. But answering your question, what are the mechanisms within the cell to prevent aggregation? I think that started already with a selection of the amino acid composition of proteins in cells. Those that were aggregating too much just didn't survive. And that's how the genetic code evolved to avoid it. And then on top of that, other proteins evolved like chaperones to make sure that they fold in the right way. There's also an interesting difference between pathological amyloids and functional ones. The functional ones aggregate much quicker to acquire the desired aggregation fold instead of while the pathological ones have more time to search through different conformations and potentially gaining those toxic ones. And one more study I want to mention here is the JAXA, the Japanese space agency. They sent some amyloid forming proteins to space on a satellite, and they found out that the protein acquires more conformational variations when there is a lack of gravity than on Earth. And there comes my question, is this another big risk factor for astronauts generating like neurological diseases because of that?
Okay, thank you.
Any other question? Okay, I had one. So it's true that you said that prions have very bad press, and they're almost universally thought as pathologic. And it was very interesting what you said about actually them having an active activity in adaptation to an environment. So I was wondering, more in a biotechnological sense, what would be the immediate applications of this?
Mm-hmm. So the one that I was thinking, there is a trade-off, for example, in biological synthesis, when we create some, for example, even referring to the startup that I just worked for, where we try to produce ammonia. The cells in biotechnology in general, there is a trade-off between division and production. So you have to reach a certain division rate and then stop dividing cells and start producing what you want. So prions, because they diversify the population and turn on spontaneously in different cells within the population, they would allow continuous production that I think it would be pretty big and bold. But in biotechnology, there is a lot of research on functional amyloids, for example, as nanowires. If you decorate protein with gold particles and create amyloid, you get almost a few atoms thin wires. Some people showed that concentrating into amyloid not necessarily has to turn off the enzymatic function, so you can actually accumulate more enzymatic function on in a smaller volume. So that's another application. But for like a prion perspective, the ability to share between different cells and diversifying population, I think the example that I mentioned about not fighting the trade-off of production and division is the biggest one that I see.
Thank you.
Thanks, Thomas, for this very, very interesting talk. I have a question from a person who knows nothing about this subject. Could you give us some more information about how is the different steps that a prion or a prion egg follows from the moment in which an animal or a person eats an infected brain or whatever? How does it get from there to the brain, and how it starts to develop there? Mm-hmm. Is this often? Does it happen very often, or how does it work?
Yes. So yeah, you mentioned eating brains. So the problem with prion disease and a mad cow disease was that the brains of other cows were fed to cows, right? So that's how the prion infected the cows. It's the theory, because it's not really known, but the theory is that the protein aggregation in the guts were the neuronal connections with the gut, and we know there is a lot of interaction between bacteria, and there is a lot of neurons in our guts. It infects the neuron in the gut and then slowly migrates up to the brain. That can take a decade. Right? And there are different types of prion aggregates, different variants of so-called variant Creutzfeldt-Jakob disease. that takes different time. There is a quick onset, there is a long term. So in general, it depends how quickly it aggregates and how well it spreads. And we don't fully understand that, but the theory is it comes to the brain through the neuronal connectivity in our body. This protein, and we're talking only about prion protein PRP, the human PRP. And this protein is also is expressed in pancreas, for example, mostly neuronal system. People think it's it's important in signaling. So,
Thanks a lot. Anyone else? No. No? Okay, I'm going to check if there's any question in Zoom. No. Okay, so I think that's it. Thank you so much.
right on time.